to this evening's live. We are going through DNA replication today. So this is going to be helpful whether you are in year 12 or year 13 if you've got mocks coming up, because this is going to be on your mocks most likely. So DNA replication, this is one of my favourite topics. I absolutely love DNA replication. What we're going to be going through is, I'll go through some of the theory as a recap, because if you've seen any of my YouTube videos, you'll know that when it comes to revising, oh, I've just realised I've got that one nail without a nail on it. Oh well, we'll have to deal with that. Anyway, what was I saying? When you're revising, you always go through the understanding stage, which is what we'll do with my notes. Then you need to test, can you remember all the information, which is what we are going to have a go at with the active recall workbook. And then you go on to practicing. So can you actually answer the exam questions? And I've got a selection of exam questions here for us to practice. So that is what we're going through in today's session. So let's start with the remembering stage. So the first thing then is just knowing the purpose of DNA replication. And DNA replication happens in interphase in mitosis and meiosis. So in the cell cycle, you always have to have DNA replicating before you can then divide the cell. Because otherwise, you're going to eventually end up running out of chromosomes if you haven't actually doubled it up every single time. So I'm just going to check this is in line for Instagram. Sorry, I'm going to pause here just temporarily because Instagram's got a little bit of a delay. And then I've just shifted the iPad. Yeah, cool. I think that's a bit better for Instagram mode. If I zoom out a bit as well. Oh, no, that zoomed in. Oh, I do this every time I go the wrong way. There we go. Hopefully that's a bit better. Okay, so yeah, before cells divide by meiosis or mitosis, you have to replicate the DNA. And the way that DNA replicates is semi-conservative replication. And sometimes that is actually asked as a 2-mark question. You're asked, what is the definition of semi-conservative replication? And if it is 2 marks, the first mark is for saying you have one strand from the parental DNA. That's the one that is conserved. And then the other one is newly synthesized. And that's why it's semi-conservative. You've half conserved, you've half kept the DNA, but don't ever say half, because half could mean it's a random mixture, and it's not. It's one entire strand is conserved and one entire strand is newly synthesized. So that's one key term, as you can see up here, that you need to know your definition of. Next, and if we have a look at the next bit of information, the process of DNA replication does heavily rely on this idea of complementary base pairs. And that's because this ensures that when you are going through DNA replication, you are creating identical copies. So make sure that you always use that complementary base pairs key phrase in your answers, particularly the complementary bit, and not just saying base pairs. And then which ones do match up. We've got cytosine and guanine, thymine and adenine. Those are your complementary base pairs. The final thing that you need to know is the role of these 2 enzymes, DNA helicase and DNA polymerase. There is a big focus on the role of those 2 enzymes. So it's really important that you do know the role of those 2 enzymes. I saw someone said, can you get these notes? Yes, so these are my A-level notes. If you go to www.missestrup.co.uk, that's where you can find my notes. And if you are actually attending any of my live masterclasses, then I'm doing a 30% discount for the day of each of those masterclasses. Each of the masterclasses is £3. So if you came to a masterclass for £3, you get 30% off my notes, which would save you something like £10 to £15. I can't remember. I can't do this math in my head. So basically you could get my notes for cheaper and a revision lesson with me. So all of that, if you just send me a message for any links that you would be interested in, you can send me a message on Instagram, and I can show you the links. But these ones are paid for because, yeah, I spent hours and hours making them. But if you do want free resources, if you go to my website, I've also got lots of resources under the freebies tab. But the ones that took me a lot of blood, sweat and tears to make, those ones do come with a price tag, I'm afraid. But yeah, this is my job, so that's why. Anyway, back to it. The enzymes. If I go to the key terms box, those are our key terms: complementary base pairs, DNA helicase, DNA polymerase. So if you did then want to make flashcards from using these notes, what I'd recommend is go to the key terms box, and that would be 4 flashcards. And then you can pick out information from the notes to make those flashcards, or from my YouTube videos. If you don't want to buy the notes, then you can still get the same information for free from my YouTube videos. Right, so the actual process then of DNA replication, those are some of the key terms and key facts that we need to be aware of. DNA replication is normally like 3 to 4 marks, and the big focus, as I said, is on these enzymes, DNA polymerase and DNA helicase, or you can just call it helicase. One thing though that I do want to point out is, if you've got the CGP textbook, there is an error in the CGP textbook, because within it, it says that the hydrogen bonds between the 2 chains of DNA are broken by DNA polymerase, and that is incorrect. That is not the case. The double strand of DNA, those hydrogen bonds are broken by the enzyme helicase. So that is the role of the enzyme helicase. It breaks the hydrogen bonds between these 2 strands and separates them. So that is the role of DNA helicase. Breaks the hydrogen bonds between the complementary base pairs. So you then get 2 separate strands which can both act as a template. That also causes the DNA double helix to unwind. Next then, step 2, as I just said, both strands act as a template. And the reason that I'm emphasizing both is when you get to transcription, you actually only have 1 strand acting as a template for transcription. So that's why I'm emphasizing that it's both strands act as a template for DNA replication. Next marking point or key concept is where we get our complementary base pairs that you need to emphasize. So complementary base pairs, so free floating DNA nucleotides, are going to align opposite their complementary base pairs. And if we just have a look at the key terms on this one, complementary base pairs again comes up. The concept of a template strand. We've talked about DNA helicase. So the last 2 key terms we haven't come across yet within this method is DNA polymerase and phosphodiester bonds. So next step then, we've got our complementary base pairs here aligned opposite each other. But what we don't have yet is they're not joined together. We need to join them together to create the new strand. I'm just going to get rid of that now. So that's then what happens. And this is the role of DNA polymerase. DNA polymerase joins together adjacent nucleotides. So what that means is it's joining together the nucleotides to create the new polymer chain. It's not joining together nucleotides, the complementary base pairs, it's the adjacent ones. And you do need to be really clear on that it's not the complementary base pairs, it's adjacent nucleotides. And that is by a condensation reaction. That is normally only worth a mark if it's at least a 3 to 4 mark question. So it's typically, the key marks are the DNA helicase breaks the hydrogen bonds, complementary base pairs align opposite, DNA polymerase joins adjacent nucleotides together. Those are your 3 standard marks for this, but you might get an extra one for a condensation reaction and for both strands act as a template. That's another one that comes up a lot. So that was the DNA polymerase. Final thing then is this concept here of the phosphodiester bond, which I'm actually going to add in here. Joining adjacent nucleotides together, let's say forming phosphodiester bonds. And now we have 2 sets of daughter DNA. 1 strand is conserved and 1 strand is newly synthesized, which is our definition of semi-conservative replication. So that is DNA replication, just a little section from my notes. And this was one way that I'd say you do the understanding stage. Just go through the information, check that you know the definitions of the key terms and that you know what the key terms are for a topic. And any places where you might want to add in more detail, like I've done here, I've said both strands are forming phosphodiester bonds, and you can do that. Last thing then, so in my notes, you do actually get at the end of each subtopic a summary of the key points and essay links. So the summary of our key points, we've said DNA replication is semi-conservative. DNA helicase breaks the hydrogen bonds. You need to know the concept of the complementary base pairs aligning, and that DNA polymerase joins adjacent nucleotides. It's a condensation reaction. And we also said phosphodiester bonds. So some examples of how you could link this to the importance of and different ideas there is DNA replication could link to the importance or anything to do with the cell cycle. So if you have something on meiosis, mitosis, or even DNA technology, so it could be a link to a theme for that for the essay, as well as gene mutations. Because that can lead to new phenotypes. It could therefore lead to natural selection, evolution, or mutations with cancer. So there's some different ways that this could link across the essays. So there's our understanding stage. Then we get to the remembering stage. And that's when I would then use the active recall workbook, is to test yourself with these short answer questions or labeling diagrams to check, can you now remember the information that we went through? And I wouldn't necessarily usually do it this close together, because I'm pretty sure you can remember something I said 2 minutes ago. Maybe not, and in which case it's going to be very helpful. But that's the concept of the active recall workbook. This is to test, can you remember? Whereas the previous one, the note, is to help you understand. Let me just see some questions that have come in. So someone said they don't get the Michelson and Stahl experiment. Oh, can you send a message to me about that? Because what I can do then is next week, I can go through the Michelson and Stahl experiment. I do have a YouTube video on it, but if you were here at the start, I said DNA replication is my favourite topic. I absolutely love the Michelson and Stahl experiment, so I'd love to go through that in a live. So remind me and I can do that. Someone also said, does this get saved? Yes, I'll post it to, it'll automatically go up on YouTube and also Instagram. So you can rewatch any of this if you want to test yourself, or rewatch it as well. Then let's see. Okay, let's have a go at this question. Before cells divide by mitosis or meiosis, all the DNA must replicate to provide a copy for the new cell. DNA replication is described as being semi-conservative. What does that mean? So this was one of the key terms I said you could turn into a flashcard. It's really important that you know this definition. It's often 2 marks. So you can have a go in the chat box. What are the 2 marks for this? And do you remember what I said is the common mistake to avoid in your answer for this as well? I'm going to start. You can have a go in the chat box as well. So mark 1 is DNA is made of one parental strand and then the second idea is and one newly synthesized strand. And the key thing that I said was you need to say one parental strand and one newly synthesized strand, with the emphasis being on the strands. That would be the key thing they'd be looking for in the mark scheme, because you can't say, I know semi means half, but you can't say half of the DNA is from the parental DNA and half is newly synthesized, because that could mean a random half. So it could be the top section, then the middle, then the top, or whatever. But it's actually one strand is parental, one strand is new. So they will insist that you say strand. So I'd say emphasize that in your notes or on a flashcard. Right, describe the process of DNA replication. So we said DNA helicase breaks hydrogen bonds. Between 2 strands of DNA. Then we said both strands act as a template. Then the complementary base pairs. Complementary base pairs align. Or in fact, it'd actually be complementary bases. To say pairs there doesn't quite make sense. Complementary bases align opposite to form pairs. But the key thing is the complementary bases. Then DNA polymerase. As the name suggests, it makes the DNA polymer. So DNA polymerase joins adjacent nucleotides, and I'm going to say here via a condensation reaction. Forming the phosphodiester bonds. Okay, so let's just highlight the key marking points for you in case you're rewatching this to create this as a model answer or as a flashcard. So DNA helicase breaks hydrogen bonds. Both strands act as a template. Complementary bases, DNA polymerase, condensation reaction, phosphodiester bond. Those will be the key things that they'd insist you have within your answer. Let's see what the next and final activity is before we go on to the exam questions. So we're told to label on this diagram which is DNA helicase. So we've got to label DNA helicase and DNA polymerase in the diagram. So we've got 1, 2, 3 options. So DNA helicase, we've said breaks hydrogen bonds. One other thing that I want to point out here that is a common mistake that people make is sometimes people say that that is a hydrolysis reaction, and it is not. Breaking the hydrogen bonds between those two DNA strands is not a hydrolysis reaction because you are not breaking apart one big molecule into two smaller ones. You're just separating two strands, and also you're not adding water. So it's not a hydrolysis reaction. And if you say that, you would lose the mark for breaking the hydrogen bonds. So it breaks the hydrogen bonds. So we can see here that is happening at this point. So that means this box, I'm going to say, is DNA helicase. I saw a question come up. What is the relevance of the 5' and 3'? It's not actually on this picture. I thought maybe you were asking that you saw it on this picture. Maybe it was on one of the others. Let me see. Oh, it probably wasn't because I drew these ones myself. No, okay. You don't actually need to know about 5' and 3' for AQA. I think you do for Cambridge International and possibly for OCR as well. What that means is which carbon within your DNA nucleotide is most exposed at the end of the chain. And it's a way to describe which end of the DNA chain you have, because you can't easily describe it. You couldn't say like the bottom or the top because the DNA could be in any orientation. So instead, the 5' and 3' is referring to which carbon within the DNA nucleotide is most exposed. And the relevance of that is because DNA polymerase is an enzyme, you can only attach at one particular end, but that is not on the AQA spec. And talk about DNA polymerase, that is this one here, because we can see it's joining together the adjacent nucleotides. We can see it's moving in this direction this way. It's joining together those adjacent nucleotides, forming those phosphodiester bonds. So that is DNA polymerase. And we have one on this side because we're going to be creating two new strands. There's also a DNA polymerase on this side. Okay, so now we've done our remembering, we've done our understanding, and now you need to go on to the exam practice. This is very similar, this question, to what we have just done in the active recall workbook. But as I've said many times in these lives, that is the whole point of one of the points of my active recall workbook is I've designed it to be a series of fill in the gaps or label the diagrams, short answer questions, which reflect things that commonly get assessed in the exam. So you're getting to the point where you're remembering the key information. So then when you get to the exam questions, it shouldn't feel as daunting and you're more likely to get more marks. And then when it gets to your actual exam, get a higher grade. So that is the whole point, that you do the active recall workbook before you move on to exam questions. So like bridges the gap between the understanding stage and the exam questions. So DNA replication is semi-conservative. Describe this process. This time it's a 5-mark question though. In the active recall workbook, we split, we did do 5. 1, 2, 3, 4, 5. Oh, cool. We already did 5. So let's just think, what do we say the key marking points were? So we have said DNA helicase. There's always going to be a mark for talking about each enzyme. DNA helicase breaks hydrogen bonds. There we go, that'd be our first mark. We then said both strands act as a template. Then we talked about the complementary base pairs lining up. So that's going to be my next one. Then we talked about DNA polymerase. And that joins adjacent nucleotides. And then we said via condensation reaction, forming phosphodiester bonds. And because I've mentioned semi-conservative replication, it's 5 marks, but I always say if you can, just give an extra one in case one of those wasn't worth a mark. I'm saying each new DNA molecule consists of one original strand and one newly synthesized. And there we go. That would be my 5-mark answer. Next then we have to create a DNA polymer. Phosphodiester bonds form between adjacent nucleotides. Describe how. So this time it's still the same process, but they've picked up or focused on a particular part of the process. So we have to say how those phosphodiester bonds form. So first thing I'm going to say is a condensation reaction. And then I'm going to, it's 2 marks, I'm going to say using DNA polymerase. There we go. Then the first stage of DNA replication is when 2 DNA strands in a double helix separate. Describe how this happens. So this time it's going to be DNA helicase involved, and I'm going to say it breaks hydrogen bonds between the complementary bases. There we go. Then if we go on to our final question, it's describe the role of DNA polymerase in DNA replication. So you can see that there's lots of very similar questions. The mark scheme is pretty much the same every time. It's slightly different wording, and it's all based on the key information we went through at the start in my notes and then in the active recall workbook as well that I've got. So describe the role of DNA polymerase in DNA replication. 2 marks. I'm going to say joins adjacent nucleotides. And I'm going to say it's a condensation reaction. So catalyzes the condensation reaction. And then it was 2 marks, but I'm also just going to say forms phosphodiester bonds in case they want that's a 1 mark and I've separated it. So forms phosphodiester bonds. There we go. And that is it. Now, before I finish up, I'm going to give you a chance just to check that because I can hear someone knocking at my door. Okay, there we go. Christmas present has arrived that I'll need to wrap up. Okay, so that takes us to the end of everything tonight for DNA replication. Hopefully you found it helpful. I'm just going to see if there's any questions. Someone just said they've just joined my membership, the gold membership. When do the live sessions take place? So you get live with the gold membership. You get 7 live lessons a month. You get 1 every Thursday doing exam questions. And then you'll get 2 AQA lessons a month, one which is at Wednesday 5:30, and you get 1 OCR lesson a month, which is Tuesday at 7 p.m. So that's if you've got the gold membership. Platinum, you get 10 live lessons a month. So hopefully you found that helpful. As I was saying at the start, people that were asking, what you've seen me use, these exam questions I've actually written myself based on AQA questions. But the bits here, these are my A-level notes. And if you do want the direct link, just send me a message on Instagram. But it is at missestrick.co.uk. This is my active recall workbook. Again, if you want the direct link, just send me a message on Instagram. It covers all of the A-level, but it's also at missestrick.co.uk. And there is 30% off my active recall workbook. And I think it's maybe the flashcards that I've done on my notes. If you're coming to my live mock lessons, there's that discount. So hopefully you found this helpful today. If you do have any requests for what I cover in these lives, just send me a message or comment on any of the recordings, because that's typically what I base what I'm going to be presenting on. So hopefully you found that helpful. And I will see you all next Thursday, which potentially, that might be the last one of the year, because the Thursday after that's the 27th, isn't it? And I'll be doing some family get-togethers then. So yeah, join me next Thursday then for the last live of 2024, which will be Thursday. So next Thursday at 8 p.m. And then after that, it will be Thursday the 2nd. I'll be back. So enjoy your evening, everyone, and I will see you next week.